At the end of the Paleolithic, 30 to 15,000 years ago, hunter-gatherers in the caves of France and Spain painted the animals they hunted and that haunted their spirits. These were mainly the mammoth, the bison, the horse, and the aurochs, the wild ancestor of the cattle that would be domesticated later during the Neolithic, roughly 9,000 years ago. Were these Magdalenian painters the descendants of the Neanderthals, the people who had already been living in Europe some 600,000 years before their disappearance 30,000 years ago? To study the biological basis of the evolution of species, both that of humans and of the animals they domesticated, a special case of human-driven evolution, the Paleogenome Group of the Institut Jacques Monod in Paris studies the genomes of extinct species such as the aurochs and the Neanderthals by analyzing the direct witnesses of their evolution, their fossilized bones and teeth. These witnesses of evolution come from archaeological sites that are well studied, have a clear stratigraphy, and are well dated. The paleogenomic data that are obtained are used to reconstruct the course of evolution.
L'ADN, si préservé dans les eaux au cours de fossilisation, est très fortement dégradé.
Ancient DNA, if preserved in fossilizing bones, is highly degraded. Only very few and very degraded small molecules are preserved. We have shown that the DNA molecules that are preserved over thousands of years are very quickly degraded after the exhumation from the sediment.
Établi des protocoles de prélèvement sur le site pour conserver l'ADN, pour le traiter comme des échantillons.
For this reason, we treat the old bones like biological samples. We keep them in the freezer until their analysis.
Et les mettre dans une glacière afin de les donner dans des conditions qui permettent la
Moreover,
conservation de l'ADN.
because of the minute quantity of ancient DNA that is preserved in these old bones, the risk of contamination with modern environmental DNA is very high.
La coupe stratigraphique. Puisque l'ADN est conservé en
To avoid contamination of the bones that we sample with our own DNA, we perform the sampling with gloves, masks, and whole-body protection suits, and, more than anything else, we do not wash the bones, what normally is a standard procedure on archaeological sites.
l'ADN qu'il porte sur lui d'être transmis sur l'os fossilisé qu'on prélève. Et c'est parti pour Paris.
To interpret the results that will be obtained in the laboratory, it is important for the group of Eva Maria Geigel to understand all the factors that could have influenced the preservation of DNA. For this reason, geologists, geochemists, anthropologists, and paleontologists all work together to discover the secrets of the preserved bones.
We go to the field not only to sample, we also try to understand how the site was built, to understand the geology, the geochemistry, the paleoclimate, the stratigraphy, briefly, all the taphonomic factors and agents that influence DNA degradation and preservation. Therefore, we study as much as possible the site in close collaboration with our colleagues from the field. This is also important to interpret our genetic results, for example, to place them in time.
Alors le site des Pradelles est un site exceptionnel justement par sa fonction.
The site of Pradelles is an exceptional site because of its function. Here the Neanderthals came to treat the animal carcasses, mainly reindeer, but also humans, which they probably used in the same way as the reindeer to extract nutritional resources. The interest of the site is that it was formed during a very cold period, the Middle Paleolithic, and, as we can see in the back, the stones that constitute the major part of the filling are embedded in a very fine clay sediment that acted as a natural refrigerator, which preserved the biological characteristics.
Les caractéristiques biologiques des vestiges osseux que l'on retrouve dans ce site.
These exceptional preservation conditions allow the research teams to study species that are now extinct. This study constitutes a major methodological challenge and requires very particular practical and technical conditions.
On dispose d'une toute petite
The extremely low DNA quantity requires detection approaches that are very sensitive, and in particular the multiplication step, that is, the amplification of the DNA so that it can be analyzed. PCR amplification is a powerful method, but is also very sensitive to contamination by environmental modern DNA. Therefore, it is necessary to perform the analysis in a high-containment laboratory and to use very strict procedures.
To avoid contamination that could lead to erroneous results, we work in a high-containment laboratory on the 6th floor of the Institute, far from other molecular biology laboratories where modern DNA is analyzed. This special laboratory uses positive air pressure to maintain a pressure gradient in order to prevent airborne DNA molecules from entering the rooms where ancient DNA is extracted and purified. This pressure gradient increases from the entrance to the innermost room where the amplification reactions are pipetted, which is the experimental step most sensitive to contamination with modern environmental DNA. Before we enter, we change our clothes and put on clothes washed with bleach that are dedicated to the work in the high-containment laboratory. Bleach degrades DNA very efficiently. We enter the airlock chamber of the laboratory and take off the lab coat that protects the clothes washed with bleach. Then we put on a whole-body suit, a mask, special bleach shoes covered by disposable protective shoe covers, and we change our gloves, which we also then treat with bleach. We also treat any object that enters the high-containment laboratory with bleach. In the first room, we prepare the archaeological specimens. This is done in separate working stations that are treated with bleach after the preparation of each sample, and with UV rays after the preparation is finished. Moreover, this working station, as well as others in the flow hoods, are equipped with a device that mixes the air while irradiating it, and produces ozone that attacks DNA. We clean the bone surface with a sterile scalpel to remove the surface that is enriched with PCR inhibitors and contaminating DNA. We then proceed to the sampling using a diamond disc that we decontaminate by flaming. The bone dust that is produced during the sawing is removed by a vacuum to avoid cross-contamination between specimens. Everything is then cleaned with bleach. The small bone fragment is then ground in the freezer mill in liquid nitrogen at minus 195 degrees Celsius to avoid heating of the molecules, which is inevitable when the material is powdered. We insert the tube containing the bone into the freezer mill, which is filled with liquid nitrogen. The bone is ground by a piston in a magnetic field. This procedure produces a very fine powder that guarantees the most efficient DNA extraction possible. We weigh the resulting powder, here 125 milligrams. We then enter the second room of the contained laboratory and we put on a second whole-body protection suit and a second pair of gloves and shoes. We then resuspend the bone powder in a buffer that contains EDTA, a chemical product that complexes calcium ions from the bone matrix and thus disintegrates to hydroxyapatite and releases the DNA molecules that are preserved in the bone matrix. This experimental step is carried out in a flow hood to avoid contamination of the samples. To suspend the bone powder in the extraction buffer and dissociate mineral aggregates, the solution is then mixed in a vortex. The incubation of the bone powder and extraction buffer takes between 24 and 120 hours and is carried out on a wheel to make sure that the suspension is well homogenized. After extraction, the DNA is purified via absorption and desorption in a silica matrix under the particular conditions of pH and ion strength. The purified DNA is then multiplied, i.e. amplified via the polymerase chain reaction, or PCR. The pipetting of the PCRs is performed in a final room with the highest pressure. Having changed the whole body protection suits, gloves, and shoes for the third time, the pipetting is carried out in glass capillaries in a flow hood. The flow hood is irradiated with UV rays at a very short distance to eliminate the contaminating DNA. During the experiment, however, the UV bulbs have to be turned off so as not to damage the skin and eyes of the experimenter. Each capillary containing fossil extract is hermetically sealed and will not be opened until the end of the PCR. The capillaries are then taken down to the modern pre-PCR lab on the 5th floor, which is separated from the genomic platform in the post-PCR laboratories. When arriving in the pre-PCR lab, we remove the clothes dedicated to the work in the high containment laboratory to put on our normal clothes and a normal lab coat. The reaction mixtures that do not contain fossil extract are then completed. The capillaries closed and transferred into the PCR machine after a centrifugation step. In the PCR machine, they are then subjected to heating and cooling cycles. This principle of the PCR allows a denaturation of the two DNA strands, followed by hybridization of small DNA primers in the strands that contain the complementary sequence. The Taq DNA polymerase, a thermostable enzyme, recognizes the primers, binds to the end of the duplex, and starts to copy the DNA strand. At the next heating step, the two strands are again separated and the procedure starts over again. The cyclical repetition of these steps allows the billion-fold multiplication of a single DNA molecule. This is the power of the method, but also its danger, since all the DNA molecules are identical to the ancient target DNA molecule we wish to analyze. These copies disperse in aerosols when the tube is opened for the following analysis steps. In our laboratory, we eliminate almost all of the potentially contaminated DNA molecules from previous PCR and cloning steps by using an enzymatic reaction that we perform before starting each PCR. Here we see in situ the synthesis of the DNA molecules thanks to the incorporation of a fluorescing molecule that allows the real-time detection of the DNA molecules when they are synthesized by the Taq polymerase. We are here in the platform of the Jacques Monod Institute. This platform contains various devices for high-throughput nucleic acid analyses and are used by the different groups of the Institut Jacques Monod, in particular by the Paleogenome group. The platform has several robots. There's a sample preparation robot and a pipetting robot for 384 well plates for high-throughput quantitative real-time PCR. Moreover, the platform is equipped with 5 quantitative real-time PCR machines.
La plateforme est équipée de 5 machines de PCR quantitative en temps réel. Ces machines,
The great advantage of these machines is that they allow us to follow in real time the evolution of the PCR amplification, and thus to determine the initial quantity of molecules, which in most cases is an extremely small number. This determination is done in a very reliable, precise, and efficient way.
petit nombre de molécules de façon très fiable, précise et efficace.
Once the PCR is finished, we see here the reaction curves of a DNA standard, a reference with a known concentration, as well as the amplification of a fossil extract that generally takes much more time to lead to a product since there are very few ancient DNA molecules. This allows us to quantify the number of initial target molecules, characterize the PCR efficiency, control the production of PCR artifacts such as primer-dimers, and quantify the inhibition of the PCR by the extracts. All these parameters are important to render the amplification of a few ancient DNA molecules more reliable. Once the PCR is finished and the curves analyzed, the products will be sequenced and the nucleotide sequences subjected to phylogenetic analyses.
À la fin du XXe et au début du XXIe siècle, l'analyse...
At the end of the 20th century and the beginning of the 21st century, the analysis of modern genomes allows to shed light on and to understand the links that relate the genomes to the morphological and biochemical characteristics of living things, as well as the molecular mechanisms of the evolution that led to modern living things.
L'homme moderne. Pour comprendre le chemin qu'a pris l'évolution,
to better understand the course of evolution, we must go back to the analysis of the fossil traces left behind and, in the case of genomic analyses, we must identify the ancient genomes preserved in these fossils. This approach, although extremely delicate and particularly sensitive to numerous artifacts, is now about to leave the domain of science fiction and to become a full scientific domain.
de la science-fiction et d'en faire une science à part entière.